the little neurons.
Are we ready?
We're going.
Okay.
The little neurons are typical shaped neurons that one would find in the central nervous system. The prototypic cell, neuronal cell. But as I said, no two neurons are identical, and biology, like English, is full of exceptions, and the very first, most important exception that we teach is that the sensory neurons that bring in information from the exterior world are special, and they have a different kind of morphology. So what we're going to make now is a sensory neuron, or a dorsal root ganglion neuron. They're called dorsal root ganglion neurons because they live in a little package that sits just outside the spinal cord on the back side of the body. So that's the dorsal side. And they're very close to the spinal cord, so they're near the root, so they're dorsal root. And groups of neurons that live together we call ganglia. So this is a dorsal root ganglion neuron, and lots of them would live together in this little package outside the spinal cord, and they have no dendrites and a very big cell body. And one process goes out to the skin or the muscle to bring in sensory information. And the other branch of that same process goes all the way into the spinal cord. And sometimes it will synapse in the spinal cord immediately, and sometimes it will turn and go towards the head and wait to synapse and end all the way at the base of the brain. So you could have a dorsal root ganglion neuron that lived in the spinal cord in the middle of your back with one process going out to the baby toe and the other process going all the way up to the base of the brain. So there's quite a lot of axon here and no dendrites, but because there's so much axon, the cell body has to be bigger because it has to metabolically support all of that length of axon. So I've got a longer string here, but I imagine I've cut way too much, so we'll just play with way too much right now. And what did I say? I was going to make a red cell body. So this time we're going to start with the cell body, and we're going to start with 2 beads for the top of the cell body. I don't know if I'm in the middle or not, but there's my 2 beads from the middle. And it looks like to anchor those, what I'm going to do is take one end and just cross thread it through. If I've done this, they won't come off. It'll be the opposite side, and I'll just have a little loop around those 2 beads, and that anchors it and gives me the top of the cell body. Right. So the next row of the cell body is going to be 4 of the red beads. Which I threaded it from one direction on one side, and now I'm going to cross thread from the other direction with the other half of the other end of the string. So when everybody gets all nicely pulled together, you've got 2 rows. All right, so now the next row is going to start to begin the nucleus. Now, in most neurons, the nucleus actually lives off to one side. It's eccentric. So I'm going to begin by making a green nucleus. 2 greens next to my 1 red, and then 2 more reds. And then I'm going to cross thread it from the other side. Keep my sides straight here. And this hemp is nice because it has some stiffness to it, but that also can work against you. As you can see, my beads flopping around. Things stay perfectly in place when you use yarn, because yarn doesn't have any of the stiffness to it. But you almost end up needing a needle to thread them sometimes with yarn. So there we go. I've got my third row. So the fourth row looks like I'm starting on the more cytoplasmic side. So I'm taking 3 reds, 2 greens, and then another red. Thread those with the second string. You want to get them just the right tightness, but not too tight that they start to buckle. All right, so this is good. All right, so now we've got one more row, and if I start on from the right, I'm going to have one. Oh, I should have had. Well, doesn't matter. I'll just make 2 greens again for the nucleus here, and then looks like a fatter cell body. I'll make 4 at this point. 3, that'll be 7. That'll be wider than it was. Neuronal cell bodies tend to be ovoid, not perfectly spherical. So if you end up making something that has a roundish biological shape but isn't perfectly round, then that's fine. That's perfectly what biology is about. Which way am I going? Go off. I got lost in direction here. All right, so let's see how that's going to look. Yeah, so now we'll cross that one back the other direction. I think you might be able to make the hemp obey later if you maybe wet it and let it dry flat, if you pressed it with something. All right, so now the next one I'm going to come and I'm going to, we'll end the nucleus. So the next will be all red, and let's make it 7 again. 1, 2, 3, 4, 5, 6, 7. And again, you cross thread with the second string. So now we're going to start making the cell body smaller again. Let's go down to 6 for the next one, next row. 1, 2, 3, 4, 5, 6. So now we're coming smaller, so the next one, let's make 5. There's an end, there's an end. 1, 2, 3, 4, 5. But again, the exact number that you use, it's not so terribly important, as long as it becomes a roundish kind of shape. And I cross thread like every row, cross thread from the other direction. Oops, 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 I got a knot. That knot comes from using string that's too long. So I have to unthread it, undo the knot, and then thread it again without a knot in it. So that's beginning to be a little oval. I think next, let's go down. We could do 4, and we could just go straight to 3. What do you think? Let's go to 3. I'm getting tired of doing this cell body. 2, 3. And again, cross-threaded from the other direction. All right, so now we finished the cell body. What comes out of the cell body is an axon. A single axon. So let's make this axon white. I've got plenty of white here. So I'm going to make a single bead coming out, which I cross from the other direction. I could make it a little longer. Let's make one more single bead. But now this axon branches. And this branching is going to be the one branch that goes out to the skin or the muscle, and the other branch that goes into the spinal cord. I'm going to keep them all the same color. But I only have one string going in each direction, so this time you can just thread on as many beads as you want to make these 2 long axons. And I'm not going to count, we're just going to thread until I get tired of threading. The colors of the beads really have nothing to do with biology. In real life, neurons are a kind of a milky, pinkish, milky, grayish, milky color. And there's no distinction in color between parts of the cell. Many of these long axons that go for long distances, either in the brain or in the spinal cord, or leaving the spinal cord or brain and going to the other parts of the body, because they're so long, the electrical signal needs to be insulated. And another cell wraps around the axons to provide that insulation. There's 2 kinds of cells that do that. The one inside the brain is called an oligodendrocyte. And the one in the spinal cord is called, or not in the spinal cord, outside the spinal cord is called a Schwann cell. And both of them produce a substance called myelin. And they wrap the myelin tightly around the axon to keep the electrical signal in the axon. They make myelin basically by using their cell membranes and just squeezing out all of the cytoplasm as the cell membrane wraps tighter and tighter around and around and around the axons. Put some on the other side for a while. Tired of doing that side. Some axons, especially those in the body wall, skin, that carry the sensation of pain are not myelinated. So they're just bare axons. Myelin helps not only to insulate, but it speeds up the flow of electrical activity down these axons. So the axons themselves don't have to be very big to go fast. Unmyelinated axons then carry signals that can go slowly. They're more slow. And even though you think you need to know about painful information quickly, what you really want is to know it's a constant, steady signal that you want to get. And so the speed is not nearly as important as the speed it takes to communicate to your muscles to move. When it's time to run, you really want to be able to move those muscles and run quickly. So that's the faster signal that you want to get down away from the brain and spinal cord and out to the body. But the sensory information coming in from touch or pressure or temperature will be myelinated. Those axons will have myelin on them. So when you're making the bead neurons, choosing colors is totally up to the person who makes the bead neuron. We just choose the colors so that they contrast. There's absolutely no biological meaning in the colors at all. The more beads you thread, the more frayed the ends get. Sometimes you just have to cut off the ends and get a fresh end. So enough for my two ends of this. So now we have two ends, and we need to make little branches for nerve terminals. Let's make these blue. So I'm going to put, say 3 beads, 4 beads on for one nerve terminal. But now I'm only working with one string at each end. So basically, I put on 3, and then I take the 4th, and I take the string around that 4th, and I go back through the other 3 blue beads. I have one nerve terminal, and I'll do the same for a second. Add 3 blues. And add a 4th that I can go around and come back through the 3 blues. Then I at least have 2 branches. And then to finish things off, I can either tie a knot immediately, or I can thread back up through several of the beads on the ends of the axon. So I'm upstream a little, and then I could go around a bead. If it's sharp enough, and if I've left enough room. It's not too tight. I've already got 2. Need to go around this one. There it comes. So I have some way. I have a second piece here that I can make a knot. All right, and then I'll just cut that off so we can dangle. I'll do the same thing at the other end. 3 beads, 1 to go around. Three beads. And let's just see if I can't make a different knot. This will just end at the end of this. Let's see if I go around the last bead. And make a simple knot there. Will that work just as well? Might be simpler. I think that's easier. Just making nice knots at the very, very end. And cutting it off. Done. So basically then we've got a dorsal ganglion neuron, a sensory neuron.